Unlocking Life's Secrets, Molecular Docking and Drug Design. Imagine unlocking the secrets of life at a molecular level. Picture tiny molecules interacting like keys fitting into locks. This is the world of molecular docking, a powerful technique revolutionizing drug design. Molecular docking uses computer simulations to predict how molecules interact. It's like a virtual laboratory, allowing scientists to test millions of compounds quickly. This accelerates the discovery of new drugs and therapies. Drug design relies on understanding how molecules bind to specific targets in the body. These targets are often proteins involved in disease pathways. Molecular docking helps us find molecules or ligands that bind effectively to these targets. This process is like finding the perfect key for a lock. The lock is the target protein and the key is the potential drug molecule. A well-fitting key can block the lock, disrupting the disease process. Exert their therapeutic effect. Preparing the key, shaping the ligand. With the target protein identified, we move on to preparing the ligand, our potential drug molecule. Think of this as shaping the key to fit the lock. Ligands can be small molecules, peptides, or even fragments of larger molecules. Researchers use databases containing millions of these potential ligands, each with unique chemical properties. Before docking, ligands undergo optimization. This involves adjusting their three-dimensional structures and properties to improve their binding affinity to the target. This preparation is like fine-tuning the key's shape and size. By making subtle adjustments, we increase the chances of finding a ligand that fits snugly into the target's binding site. The dance of molecules. Docking in action. Now comes the heart of the process, molecular docking. Here, the ligand and target are brought together virtually to simulate their interaction. Imagine it as a delicate dance between two molecules. The docking software uses sophisticated algorithms to predict how the ligand will bind to the target. It explores various orientations and conformations, searching for the most favorable binding pose. During this virtual dance, the software calculates the energy of each pose. The lower the energy, the more stable and likely the interaction. This energy calculation helps us rank different ligands based on their binding affinity. Through this intricate molecular dance, we identify ligands with the highest potential to bind effectively to the target and exert a therapeutic effect. Section 5. Finding the perfect fit, binding affinity and scoring. After the docking simulations, we need a way to evaluate the results. This is where binding affinity and scoring functions come into play. Imagine them as judges evaluating the performance of each ligand. Binding affinity refers to the strength of attraction between the ligand and target. A higher affinity suggests a tighter binding and potentially a more effective drug. Scoring functions are mathematical algorithms used to calculate a score for each docked pose. This score reflects the predicted binding affinity and helps rank different ligands. Like judges awarding points for different aspects of a performance, scoring functions consider various factors. These include electrostatic interactions, hydrogen bonding, and hydrophobic interactions between the ligand and target. Section 6. Exploring possibilities, conformational space. Proteins and ligands are not static entities. They are flexible and can adopt different shapes or conformations. This flexibility adds another layer of complexity to molecular docking. Imagine the ligand as a dancer with multiple moves and the target as the dance floor with various grooves. The ligand explores different conformations to find the best fit within the target's binding site. This exploration is crucial, as even slight changes in conformation can significantly impact binding affinity. Docking software must account for this flexibility, searching through a vast conformational space to find the optimal binding pose. By considering the conformational flexibility of both the ligand and target, we can make more accurate predictions about their interactions and identify the most promising drug candidates. Section 7: Tools of the trade software for molecular docking. Performing molecular docking requires specialized software designed to handle the complex calculations involved. These software packages are the tools of the trade for researchers in drug discovery. Popular molecular docking software includes Autodock, Gold, and Glide. Each program uses different algorithms and scoring functions, offering a range of options for researchers. These software packages provide user-friendly interfaces for setting up and running docking simulations. They also offer visualization tools to analyze the results and explore the interactions between the ligand and target in detail. The development of increasingly powerful and sophisticated software has been instrumental in the rise of molecular docking as a vital technique in drug discovery. Section 8: From Screen to Synthesis, the drug discovery pipeline. Molecular docking is not a standalone solution, it's part of a larger drug discovery pipeline. This pipeline involves multiple stages, from identifying a target to developing a marketable drug. The docking results guide the selection of promising ligands for further testing. These ligands may undergo chemical modifications based on the docking insights to improve their properties further. Promising candidates then move on to experimental validation. This involves synthesizing the ligands and testing their activity against the target protein in vitro and in vivo. The entire process, from initial screening to developing a marketable drug, is lengthy and complex. Molecular docking plays a crucial role in accelerating this pipeline by identifying potential drug candidates more efficiently. Section 9: A future of targeted therapies. Molecular docking holds immense promise for the future of drug discovery. By enabling the design of more targeted therapies, it paves the way for more effective treatments with fewer side effects. As our understanding of disease pathways and the availability of computational power increase, molecular docking will continue to advance. This will lead to the development of even more sophisticated software and algorithms. Imagine a future where personalized medicine becomes a reality. Molecular docking could enable the design of tailor-made drugs based on an individual's genetic makeup and specific disease characteristics. The journey into the microscopic world of molecular docking is an exciting one, filled with the potential to revolutionize medicine and improve human health in countless ways.